water, and it's not a good feeling. Okay, so we need to think about hormones again. And I think we left off around here. So we've been covering like they call them the 5 major hormone groups. And ethylene is just ethylene, abscisic acid is just abscisic acid. All the others groups of hormones, they definitely have many different types of auxins and gibberellins and cytokinins. But abscisic acid, even though it's just one molecule here, it is very, very important for the plant. And especially when it comes to water stress. And we thought about this last time where if the plant is the typical plant with its roots buried in the soil, it's the roots that experience drought first. That's the first part of the plant body. And so that's where abscisic acid is made. It is made in response to that lack of water. But the issue is to be effective in your response to that lack of water, the main thing you should do is shut your stomata, which of course is way up in the leaves. And so you have to get that abscisic acid that's being synthesized in the roots all the way up to the leaf tissue and then make it specifically to the guard cells and have the guard cells do their thing to close over the stomata, the pore. And so you have this process. It happens really quite quickly in terms of experimentally when they put that plant under drought conditions. The abscisic acid gets synthesized very quickly, moves very quickly through the plant body to exert its effect on the stomata. And so here's an image trying to get at that visual where it's the roots going through the drought. They're like, eek, I'm getting low in water. And so they will synthesize the abscisic acid and get it sent up. And then here you see the idea of targeting the guard cells. So we have this situation where, let me go forward one slide. So it's an interesting situation because abscisic acid will come out of the xylem in the vein. And if you have a well-watered plant, it tends to go more into just like the spongy mesophyll cells and just have that impact. But if you have a water stress, it actually shifts the pH from slightly acidic to more alkaline under water stress. And that little shift allows for more abscisic acid to get routed to the guard cells. They're less likely to be taken up by any of the spongy mesophyll. And so it's just this little pH shift that's allowing for abscisic acid to reach its destination under drought conditions. So again, pretty simple mechanism, but in the grand scheme, there's a lot going on here, considering abscisic acid is made way down in the roots and has to get to the stomata. So they've done lots of experiments with this, and here's some data showing if you withhold water. So up here, you see the water is being withheld and you see the water potential in the leaf starts to go more and more negative, more and more negative. So that definitely is saying that they're under some water stress. And then if you match that up, if you follow the dotted line down to this lower graph, you can see stomatal resistance is in the black line. What they mean by that is how much frictional force is being measured through the stomata, the pore. And so if you get a lot of resistance, that means the pore is very closed down. And so there's more friction on the air molecules and water molecules coming through there. And so that's why you see this black line, it's swooping up where you have greater and greater resistance. That just means it's closing down, the stomata is closing down, and then it matches the abscisic acid synthesis and concentration matches very well to that idea of the stomata start to close down as you get this abscisic acid rise as well. So they have some nice data to support this idea of linking abscisic acid, increase in abscisic acid, and then the stomata closure when you have drought conditions. And then they went ahead and said, okay, let's water the plant really well. and then you kind of just see the reversal of everything. You see less negative water potential. You see the resistance going down, so that means the stomata are starting to open up again. Abscisic acid is no longer needed, so that starts to decline as well. And so nice data showing that under certain conditions, yes, you definitely need hormones upregulating, get higher concentrations, but then at some point you're going to need that to quiet down. And after the emergency, in this case, the drought emergency is over, so go ahead and quiet down the abscisic acid. Okay, so a couple of other categories I just wanted to mention. Steroids are relatively recent. This is a newer discovery compared to all the other ones we just went through. And they're still working out the details of what it can do for the plant, but they do know it's similar to steroid hormones in humans and other animals. So if you've ever studied that in animal or human phys, that the structure may look vaguely familiar to those steroids. What they're saying right now is, well, we think there's wide effects on the plant. So they see that steroids are involved in the cell division and elongation process. You got germination and senescence, stress. So, I mean, these are all big ticket items here. This is really important stuff that the plant needs to regulate and deal with. And even the sex of the flowers, they're finding the steroids are involved in getting proper flower formation. So some flowers, as you know, some flowers that might be just male flowers and separate female flowers. And so here, I don't know if you can tell maybe if you look right here, can you tell what kind of plant this is? It's a little hard. It's supposed to be a corn plant. It's supposed to be corn on the cob idea there. If you look at it and squint, you might see that idea here. So if you've ever gone and picked corn from corn plants in a field, that's the female flower. It's actually many flowers stuck together in a very tight pattern. So it's many female flowers all clustered. And all that silk, those long strands that you end up, when you shuck the corn, you peel away the leaves covering the corn and then you get rid of the silk. The silk is the female pistil of the flower. That's what catches the pollen and allows the pollen to eventually get down to the ovary of the flower, which then turns into the kernel that you bite into when you eat corn on the cob. So all of this kind of stringy looking stuff, that's silk-ish. And so what they did here, and on the left image here, that is supposed to be the male. But you're seeing it's just messed up. This is not what corn plants should look like. And so they were playing around with making mutants of steroid hormone plants, the corn plants like that. And the wild type is on the left. That's what the staminate should look like. So when you look at a corn plant, you have the female flower a little further down on the stem of the plant, and then the male tassels are up high. And so this plant is supposed to be the female, but it's not normal. Let's just put it that way. So this is something that they're playing around with, trying to figure out how the steroids are regulating proper flower development, when you do have these separate flowers, male flowers versus female flowers. So it's just not normal if you don't have the steroids doing their thing. I'm going to mention these two later, so I'm not going to say much about them right now. Salicylic acid and jasmonic acid. They are, again, fairly newly discovered in plants, although actually salicylic acid is related to acetyl salicylic acid. Does anyone know what that is? You might have taken some through your life already to help with headaches or something. Acetyl salicylic acid, say that 5 times fast. That's aspirin. And so salicylic acid Jasmonic acid. So I'm reserving chatting to you about those because they're involved heavily in plant defense mechanisms, and we're going to cover that soon. And so I'll save it for that. And you'll see a little bit more about how they interact, but they have found that they're very, very important in plant defense against pathogens, and then also dealing with abiotic stress, which we'll also cover separately here in a few weeks. So hold on to that thought and we'll get back to those. And then probably one of the more recent discoveries out there are strigolactones. These also, they're interesting. The more they look, the more they find it. Now they're hypothesizing that, the majority of plants seem to have strigolactones. And the reason they're called strigolactones is that they were discovered in a parasitic plant. The genus name of that plant is Striga or Striga. I say strigolactones, some people strigolactones, it just depends. So that I'll mention a little bit more about Striga. But what they're finding is that this hormone does seem to suppress branching out of both the stem tissue. So it reduces branching of shoots in the stem tissue and also roots too, that they don't have a lot of lateral branching roots off the main root. On the other hand, it seems to stimulate root hair growth, which is really important. Plants are continually having their root hairs damaged just through normal growth and wear and tear. Root hairs are kind of fragile, and so you need a constant replenish of that. And then one of the other main areas of research on this particular hormone is that they know that it's involved in forming a partnership with VAM fungi, so the vesicular or buscular mycorrhizal fungi. The plant will excrete out the strigolactones to advertise for the mycorrhizal fungi to find them and set up a partnership. And so they've been looking at that pretty heavily as well. But initially, they didn't know that it was a broad plant hormone until fairly recently. They knew about it, but they thought it was only for parasitic plants. So that's what you're seeing here. On the left, this cute little plant with these beautiful little red flowers. That's Striga. And what's happening here is it's living right at the base of a corn plant in an agricultural field. And so this corn plant has this Striga right there, right next to it. And so what you're seeing on the right, this big cross section here, that's the corn stem. And then this blob that you see on the lower portion of the picture, that's the Striga plant. And what it's doing here is it's plugging in, they call it a hostoria. You don't need to know that. That's just a specialized structure that parasitic, well, not just parasitic, any organism that creates a special like intrusion mechanism to tap into, in this case, the corn plant. This is more of a parasitic relationship because they know that Striga is sucking away the nutrients from the corn. And so in order to do all of this, in order to hook into the corn stem like that, strigolactones are involved in that process. And so Striga is making the strigolactone to become a successful parasite, not good for the corn plant. And so if a corn grower starts to see this in their fields, they're dismayed because this is a really big problem. It can cause significant biomass loss and yield loss of the corn plant. And it's just really hard to get rid of once they set seed, and then the seeds fall to the ground and become part of the seed bank of that soil. Very challenging to get rid of. And so they've been struggling with this idea for quite some time, and they're trying to work on ways to eradicate Striga, especially, of course, out of agricultural fields. So it's this tiny little cute plant, and yet it causes a lot of economic hardship on people that are trying to grow food for food production. So that story is something that's continuing on, and hopefully they'll find they're trying several different tactics to deal with that. But It's probably going to take multiple different avenues of attack, so to speak, to get that resolved. But here again, this image is trying to get you to think about different ideas of how hormones become active and like the different avenues for that. So on the left, the green boxes are trying to show that they can take up a hormone, they can release it from maybe the vacuole if it's being stored there. There's activation ideas where maybe it's conjugated and they have to cleave off a sugar or something to get it active. They could synthesize it from scratch. So just trying to get you to think about a lot of this we've already talked about in the sense of you have to get an active hormone up and running. You might quiet it by conjugating it, but then it's quickly released. All of this on the right is just saying, okay, once that signal transduction pathway has run its course and that's like no longer needed, you have to have a way to quiet everything down. And so that's trying to get at that idea. Efflux just means you can get rid of it out of the cells and tissues. Break it down, conjugate it again, store it away. Sequestration, maybe you store it away in the vacuole. So they're just trying to show that you have to have this balance of, yes, you have to have signal transduction going on. Yes, hormones are important and they have to be up and running, but at some point you got to get them to be quiet. And there's lots of strategies for that, depending on the plant and the tissue and the hormone. It's complicated. So in general, if you think about hormones that go through the plant body, where they have to, let's say they're made in the shoot tip, but they're needed further down, maybe at the base of the stem or something. So you got long distance travel. You can think of, there's different ways that you could have that happen. How do they get from point A to point B in a multicellular organism, and sometimes a huge organism? Some plants are huge. You could go cell to cell, and some hormones do that. But that tends to be a little bit more of a process. So you could do it through diffusion processes where the hormone moves from a higher concentration to a lower concentration. They can see that happening when they look at it experimentally. They can find transmembrane proteins for many hormones where the hormone is going through the protein. So they know plants do this. They have some of those channel proteins. Sometimes experimentally, they do see that active transport is needed to get movement of the hormone from cell to cell. I'll mention that with auxin in a minute here. So active transport, remember that means ATP. You're putting in the use of ATP. So it's an energy cost. So you could have that kind of movement, and experimentally they show that can happen. Tends to be more for short distance travel, which kind of makes sense because this is a little bit involved if you have to go through a bunch of membrane proteins, and it's just a little bit more cumbersome. The quicker route, if you're going to really go long distance, would be course your way through the xylem or the phloem, and they do see that as well. So when they analyze content of what's in the xylem, yes, there's definitely water. Yes, there's definitely minerals, but depending on the situation the plant is experiencing, hormones can also be in there. Same with the phloem. And so you do have methods of movement kind of through that internal highway system of the vascular tissue, which makes sense. But let me just mention a little bit about these transmembrane proteins. Let me give that example with auxin. So here you have 3 cells, neighboring cells, and they're trying to show here the polar transport. So that's the idea that auxin, that if it's synthesized in the shoot tip, it will move down to the base of the stem, no matter what, if you tip that plant upside down, it's still going to do that kind of movement. And so this polar transport, so to speak, they're showing up top the indolacetic acid, so that's your auxin. And they're trying to show with those pink balls that those are the transport proteins that are allowing the auxin to come through. So they're embedded in the cell membrane. And then they're trying to show that it's coming out of that cell and going into the neighboring cell again through another transport protein. And you can see active transport is involved because they're showing that oxen and a proton are going in together. So that is implying you had to get the use of ATP to get that proton gradient. So that's what they're showing here. ATP is being used to push protons out, and then it can come and help oxen come through. All right, so this is the symport idea where you have two molecules coming through that transport protein. In this case, it's a proton and the oxen. Okay, so they're trying to demonstrate a lot of things here on this diagram with the use of active transport, get your proton gradient, then use symport processes to pull oxen into that next cell. And then they're showing movement of oxen through the cell till it gets to the other side. And then you see again, it's going out. Whoops, it's going out of that cell and it's making its way to the next cell. So it's kind of involved. And it really does kind of make some scientists go, why are they choosing that method? And they do choose that method sometimes. They don't go with just the xylem or foam transport. And so that's led to some interesting hypotheses, which I talk about in my advanced botany class, but especially when you get into the root zone, auxin is produced in roots too and can flow from the shoot to the roots as well. And they have some interesting ideas about that, but it's kind of makes you scratch your head that they have this pretty involved process. Okay. So we have cell to cell, xylem, phloem. So hormones definitely can be transported through all these mechanisms. There are other things going on though too that plants use as a way to transmit signals, and that's through electrical signaling. And it can be something very localized, like with the Venus flytrap here. So it's just within those modified leaves that you get this very relatively quick electrical signaling going on there. That's an example of a fairly quick response through electrical signaling. And so these trigger hairs that you see, I think I might have mentioned that briefly the other day. You can see that there's many trigger hairs. There's actually more than what they've shown here, and they must have amputated some of them. But anyways, if an insect is crawling around in that trap, it's going to trigger more than one trigger hair as it's moving. And that's the signal to the plant to clamp shut. If you can imagine a stick or a piece of debris landing in there that's not going to move, then it won't snap shut because it's not getting the signal that there's something moving on my leaf. And it's probably not something that I want to eat. And so they've done those types of experiments in the lab. And so this is one reason why they say plants can count, because the plant has to count two trigger hairs being touched in succession within, I think it's 10 to 20 seconds. And if that happens, they'll close. If that doesn't happen, they won't close. And so they're closing rapidly because of an action potential. And I'm not going to go into detail on this, but just be aware electrical signaling is a method of transmitting a signal and to get the plant to do an appropriate response. And it can be very localized, but it can also work long distance. And I'll mention a little bit more about that when we get into plant defense, because they're doing that sometimes when they are being attacked by a pathogen. They'll send electrical signals through their body to get their whole body to prepare for attack. But if you're wondering what this really is, I'm not going to ask you to tell me this on an exam, but with electrical signaling, it's a transient depolarization of the cell membrane. generated by voltage-gated ion channels. So if you've taken a neuroscience class, that may sound vaguely familiar. That's what they talk about when you think about how our brain works. It's through that idea. So plants are capable of doing a very similar thing as what we do with our brain when we're sending signals from neuron to neuron. So they're looking at this more carefully with electrical signaling and what roles it plays. And the more they look, the more they find. So it's really interesting research. I'll talk a little bit more about this, like I said, when we get into plant defenses. Okay, so now the next set of slides. They look pretty busy, but I'm going to try to chunk them out for you and show you they're not as bad as they look. And I'll point out specifically what I want you to capture from these slides that are, these images that are coming here. So when you look at the signal transduction process, when you look at how plants are doing this, they actually ripped off how bacteria do it and took that as a model and decided to go with it, which was a good thing. And so through the evolutionary process, bacteria were the first things on the planet. They were able to get sophisticated and start getting signal transduction mechanisms in place. And they do it through what we call a two-component signal transduction pathway. So I'm going to break that down. Two components. That means there's two major things to think about in this signal process. So this is the first one. There's a sensor kinase protein. It's the sensor. It's the one that will pick up on that signal, maybe a hormone docking into this. So this is a protein. It's a receptor protein. So it could be a sensor kinase for a particular hormone. So that's one of the players of the two component. The second one is the response regulator protein. Okay, it's just another protein. It's in the cell, typically, not always. Sometimes it's in the cell membrane. But anyways, it is going to interact with the sensor kinase in some manner. Maybe not directly, but somehow the sensor kinase has to relay, okay, like maybe it's cytokinin that docked into the sensor kinase. And then it's got to relay, I got a cytokinin here. We got to start, moving on this information. And then the response regulator will get that signal next. And then it's the job of the response regulator to further get that signal transmitted, typically all the way to the DNA, and start shifting or altering gene expression. And so the typical way that this happens is they use a phosphate or a phosphoryl group as a method of sending the signal further into the cell, transducing that signal. So your body is doing this right now. You have this phosphate transfer system going on too when your cells and tissues are doing the signal transduction thing. So it's not unique to plants or bacteria. We do this too. So really, it's a pretty elegant way to be able to sense something going on in your environment and then respond appropriately. And you got a couple players in the game, plus a phosphate group that gets transferred to further send that signal. I often think about running relay races where you have the baton, you run with the baton, and after you've done your lap, you give off the baton to the next runner in the race. That's kind of what I think about when I'm imagining. So the first runner in the race is the sensor kinase and grabs the phosphate, that's your baton, and run, and then gets back to the starting line and gives the baton off to the response regulator, and then they got to do their job. Now, instead of running, what they're doing is getting that signal, that initial signal, further transduced within the cell all the way ultimately to the DNA. So plants can alter gene expression appropriately. Maybe it's a signal, go ahead and grow your stem. You need to grow taller because you're getting shaded out. Maybe that's the message. And so the plant goes, oh, okay, we gotta get behavior to respond appropriately to that hormone. It's gibberellin would be giving that information to grow taller. And then the gene expression would shift to stimulate stem growth. So the left image is what we see in what bacteria do. All right. And so their bacteria have to constantly assess their environment as well, just like plants. So they're using that phosphate group to transfer from the first component, which is the sensor kinase, to the second component, which is your response regulator. Now, if you compare that to what you see on the right, it's pretty much the same thing. They just added a middleman, middle person, so to speak, to this whole thing. Okay. And the proteins are a little bit more sophisticated. But it's the same idea. You have a sensor kinase that will interact with some signal that it's getting, and then it's going to transfer a phosphate. And in this case, it goes to the middle, the transfer protein in the middle, and then that one gives it to the response regulator. Okay. So they just added a step, but it's the same basic idea. Transfer that phosphate from player to player in the signal cascade. And then the ultimate goal is to respond appropriately to whatever that initial signal was. So that typically means you got to shift your gene expression to respond appropriately. So again, if you kind of just step back and look at what is really happening here, it's not as complicated as it looks. Now, here are some examples of, okay, like what's the signal? So I'm going to break these down. These next images are just blow-ups of what's on this slide. So let's look at this one. And again, it's this idea now, here you have your hormone, cytokinin, and it's docking in to the sensor kinase. Okay, so all of this is your sensor kinase. So now the sensor kinase goes, whoop, I got a cytokinin. And then that activates the sensor kinase to hand off its phosphate to the next player in the game or the next runner in the race. And then you see they just kind of truncated it and said, yeah, and then it just keeps going. You have a signaling cascade. And then, eventually the gene expression will shift in some way. So they're just kind of zeroing in on the first steps here. So that's an example. Here's with brassinosteroid. Here's a little different where you have two proteins. The sensor kinase is in blue here. And then here's your so-called target protein. And now they show the brassinosteroid interacting with the sensor kinase. And then that transfers a phosphate group. So again, with that docking of the hormone into the sensor kinase, that allows for the phosphate to get handed off. And then they say, and then it just keeps going until you get ultimately to the DNA. Here again, this is abscisic acid. So again, you see once abscisic acid comes in, it docks with the sensor kinase, you have a phosphorylation event. It's a very common strategy. It doesn't matter, which hormone we're talking about, you still get this relay of phosphate from player to player in the signal cascade. Does that make sense? Okay. So I'm not expecting detail on these images. It's more the big picture idea that the two components, that you have a phosphate in order to send the signal and keep it going into the cascade process. And then I would like you to know that ultimately it's at the level of DNA that you usually get the ultimate, shift in behavior. You'll get a shift in gene expression so that the plant can respond appropriately to whatever the initial signal was. Now, these next slides, again, this looks really busy. And again, I'm not going into huge detail. There's a couple of big picture ideas off of this that I want you to get. This is looking at the idea, I put it in yellow up here. Signal transduction pathways in plants often function by inactivating repressor proteins. I would like you to know that. Okay, so what does that really mean? What we're thinking of is at the level of the DNA. So that's what you see here. This is DNA, a DNA strand, okay? And you have this weird looking kind of peach colored, I don't know what color that is, tan, pink. That's your repressor protein. What happens is it's repressing gene expression. So it's keeping gene expression quiet in this section of DNA, in the blue section here. It's keeping it quiet. Okay. Well, then auxin comes in. So auxin, in this case, will bind and it essentially, part of what's happening here is the repressor protein gets removed off the DNA. And a lot of what's on this slide, they're going into details of how that happens. Basically, they're showing that the repressor protein gets degraded, gets broken down. That's the upshot of it. But basically, when you look at it, the presence of the auxin causes that repressor protein. They're trying to show that the repressor protein is getting chewed up. That frees up that space where now a transcription factor, another protein, can come in and turn on that section of DNA. So, yeah, there's a lot of detail on here, but the upshot is often you have a repressor protein making a section of DNA really quiet, but then the presence of the hormone comes along and creates a situation where that repressor protein comes off or degraded. And then that frees up the space for the proteins that control gene expression to come on in and start doing their thing. In this case, they're turning on gene expression in that area. They're doing transcription. So I'm not getting into the detail of exactly each process. So these are 3 different examples with auxin, jasmonate, and gibberellin. And then these are blow-ups of that slide. This is just auxin, so that's the one we were focusing in on just now. And then there's jasmonate. But again, it's the same basic idea. The presence of the hormone ultimately gets rid of that repressor protein. And then that section of DNA can get turned on. So that's the idea of inactivating repressor proteins. You have that repressor protein going away. And so now it can no longer repress that section of DNA, and now it can be turned on. So that's a common strategy. It's another way that hormones can interact with the cells and make sure that the signal is definitely heard and they respond appropriately. And then his last slide. Ugh. So this last slide, I won't go into it, other than this is just showing the idea that you have lots of strategies. And all I'll say about this, whoops, my chicken scratching here. It's complicated. Right, so here you're looking at signal transduction pathways that can start to hook up with each other. And so you can get really complicated signal transduction pathways. And my hat goes off to the people who figure all this out, because this looks like it could be very, very challenging. So they know that there's primary interactions. Basically, I've been talking to you a lot about that. And then there's secondary and tertiary interactions. I'm not going to ask you about this slide other than I'd like you to realize that it's very complicated, and that what I've covered in this slide set is kind of the big picture idea, and there's a lot more to it. So This is a very active area of research, but still a lot to try to discover because you have these signal transduction pathways linking together and doing all kinds of interesting things, positive interactions, negative interactions. But basically, all of this is in place and has evolved so that the plant that can't move away, can't run away, it has to have a very sophisticated mechanism, many mechanisms in place to be able to constantly assess what's going on around it, be able to take in that information appropriately, and then figure it all out and then respond appropriately. That's a huge undertaking when you, especially when you think about an organism that can't move away from where it is. And so they are very sophisticated in being able to be very quickly adaptable to what's going on around them and respond quickly. Plant physiologists say more so than humans and other animals, because humans and other animals typically can just move away from a situation and don't if they don't like it. So, okay. So yes, definitely complicated. All right. So that slide set was talking a lot about this, how do the signals work, including the hormones as signals? How does that all work to do this signal transduction? So now I'd kind of like to come a little bit more back off a little bit and talk a little bit about some of the ideas that they have about using hormones in an applied way. So this is more to do with in real world applications, how have people been using some of these hormones? And so of course, this all came after they figured out plant had hormones to begin with and what they're doing for the plant. And then they asked the question, how can we use that information to benefit, what we want from plants, which often is food production or material for, building homes or building materials in general. We use plants a lot. Most people are kind of blind to that, but they're such an integral part of our lives. So I'd like to talk about some ideas that have been in play for a while now and have been effective. So auxins, we know that auxins stimulate plants to grow, but if you overdo it, they can kill plants. Okay, so many hormones are dose dependent. Too much of anything can be bad for you. And so 2,4-D, you might have heard of 2,4-D as an herbicide or dicamba. Those are synthetic auxins that get applied to the plant fields at higher doses, and it can be effective at killing off plants. Some plants are more sensitive than others to higher doses of auxins, and that's why you can have some plants killed and others in the same field not harmed. So they have that level of understanding when they apply these types of treatments. Here's something you can do to your houseplant. So one thing that they discovered is that the presence of auxins in the shoot tip provides a situation that they call apical dominance. So what they mean is that if the shoot tip is intact and if the shoot tip is producing auxin, it gets synthesized and then it flows down the stem. And what it does as it's flowing down the stem, it tells the axillary buds, which are further down the stem at the node regions. You have axillary buds. Whoops. Okay, there's axillary buds right at the node regions that are also meristematic. They can start to get activated and send off branches and leaves. Okay, so what's happening is at the shoot apex, under many stretches of time of that plant's life, those axillary buds are quiet. They're not doing much. And that's because of apical dominance, the auxins flowing down. And it's telling those axillary buds or lateral buds, just stay sleeping, chill out, don't do anything. If you experimentally, or you can do this to your house plant, if you don't like how tall your plant is getting, you can take off the shoot tip. You can amputate it. That's what they did here. So this is, they amputated the shoot tip and then they waited to see what would happen. Now that auxin's gone, no more auxin flowing down they really amputated it far down. You only have a little section of the stem there. And now you see those axillary buds became active and sent out more leaves. So if you have a plant at home that's getting too leggy, too long and skinny, and you don't like how it's not really getting fleshed out, so to speak, you can do that. You can pinch off or cut off the apical meristem, and then that relieves apical dominance. Now the axillary buds go, oh, I'm not feeling any auxin bathing me, so I'm going to wake up and start growing. And that's what they did. And that would change the shape of your house plant. So I do that often. A lot of my windows are not good windows for sun exposure in the house, so they don't get enough light, so they tend to get very leggy as they're constantly searching for better light. So that is one thing I do. They also know that auxins are good at getting the lateral roots to form. And here they're showing a situation where they generated a mutant that produces more than its normal amount of auxin, but not toxic levels. And it's really going crazy on lateral roots compared to the one on the left. So if you've ever used rooting compound, if you get cuttings of plants and you take a stem cutting and you want it to start rooting, if you dipped the stem into that powder, or sometimes it's liquid, that rooting compound, it contains auxin, or probably indole acetic acid, that version of auxin. And so it's stimulating the cut area of the stem to go ahead and start producing roots. And so that's one of the things that you find when you go and buy that commercial rooting compound. Okay, we're going to continue on. There's other things. I'm going to talk about the big 5 groups of hormones and how we can use them in real world, but we'll stop there for today.